they're done. But in the meantime, we're going to be looking at a beautiful pink coral here.
Is this Paragorgia?
I don't think so. I think it's going to be a hemicorallium in the Coralliidae
Oh,
group.
precious coral.
Precious coral.
Excuse me.
These have a hard skeleton, but they're not hard corals, which are generally, that term hard coral is usually denotes members of the Scleractinia group. This is in the family Coralliidae. It's an octocoral, but you can see this very sort of hard branch structure that it's got, and shallower members of this family are harvested in some places of the world, and they're used to make jewelry. A colony like this, however, could take 100, 200 years to get here.
Okay, there's an associate. Or no, that's another coral behind it, I think. What is that?
It looks like we have a Chrysogorgia behind it.
Ah, yes.
Right. It's probably one that we have already imaged, so we're good, pilot. Thank you.
Let's get the whole thing, and then we can be off.
Turn to starboard just to see if I can get eyes on that sonar return.
Okay. Let's get lasers for size, please.
Lasers on.
And that's a dead sponge next to it, by the way, that's toppled over.
Okay,
the brown, large, weird structure.
Full wide, please.
So I've mentioned this before, but in case you're just joining us, when one of these glass sponges dies, and if it remains attached to the substrate, it can collect a lot of sediment. And that's what I think makes them, turns them brown.
Chris, to follow up on this question about why we see those sponges settling on these other ones, there are the different attachment points, right? What was the name? Phytophagus? I forget the name. You'll have to remind me about that. But
Yeah.
is it possible that that attachment type is not suited for whatever reason, the bottom that we're looking at here?
Yeah, so Scott, this is manganese-crusted boulders, as we've seen in a lot of places here, and I think we can go ahead, pilot, and just continue on up slope. So we've seen a lot of these sponges that seem to have no trouble grappling onto the seafloor here. Its Lophophytus attachment is the one that grapples with modified loose spicules that are called basalia, and then Basiphytus attachment is actually where, like this white sponge that we're seeing right now, that these other ones are landing on, it forms a plate and glues itself to the substrate. And I've noticed in some places, at least, that the grappling system seems to actually be a much sturdier attachment. Oftentimes, the live part of the sponge is long since dead, yet you can still see that lower part grappled onto the substrate. But now you're seeing these upright sponges that look pretty darn glued and upright too. So I don't know.
Sim?
Right, and so I didn't mean to suggest that it was the manganese crust itself, because I agree, we see that fairly routinely.
All right, co-pilot, the ship should be coming to a stop.
We don't know, but to me that's the difference here.
D2 USBL has been kind of crazy for the last 2 minutes or so. Do you want me to stop it?
Although no, didn't we also see Bolosoma, I think? So that would have the same attachment type, right, as these ones?
Right. The Bolosoma and these Spidoscopulia, which I think these other standing ones are, they are Basiphytus, so they glue. So, so anyway, it's a,
Thank you,
it's a very curious observation, so it's quite interesting. Yeah, thank you.
I see a couple more coming up on the distant left there, are still on the sponges instead of on the bottom, so very curious. Love seeing these new observations.
Look at that. There's another one. Look, you can see them uphill.
That's really weird.
This is very weird. It's kind of fun, though, because it's obviously generating a lot of speculation.
Let's look at what kind of crab is on this dead sponge stock to the left.
All right,
Or squat lobster, I suspect. I think I saw another one crawled up on top of.
by
that's
the
it.
lasers, yeah.
Yeah.
Okay, lasers off and okay to zoom.
Alright.
Aha.
That's a partial.
Okay. Well, we got this to genus, but I'm not sure I can go any further. So this is Munididae. I don't know if I can say this right. In the family Munididae. And sorry if I butchered that. And I believe this is in the genus Munidopsis, but I don't know if others have a different opinion. They've got very, very long spines sticking out between his eyes. As you can see, he kind of looks like a deep sea rhinoceros.
Presumably, they're like a lot of the other animals around here. They're taking advantage of the current flow to feed. Crawling up on all of these sponge stalks is a great way to do that.
A little bit more zoom. Yeah, that's good. We'll just get this in the can and move on. And confirm lasers off. I think they're still on.
Almost looks like something we call Munidopsis bairdi, but I'm just
Sorry, I was
gonna
muted.
wait
Lasers
on that.
are on. Would you like
bet
them off?
Mary Wickstein
Off,
will email
please.
me in a
Off.
couple days when she gets a look at these photos. She's at Texas A&M and helps out a lot with the arthropod IDs, and I'll bet she'll know.
Indeed, Mary is very knowledgeable in these subjects.
Okay, video, we can pull out, get the whole sponge, and then I'll be on my way.
Sounds good.
A couple crinoids there to the right.
Picturesque there. And you come full wide when you're happy.
Alright. And we're full wide.
Alright, off we go.
I think we need to find us some sediment.
Good luck with that today.
Yeah, I know.
See if I can muddle my way there.
Oh, no. No. Hopefully you guys didn't hear that. Dave's joke. This stock sponge here to the right center, this is something different. We haven't seen this today so far.
Near ground? Far
Yeah, right center right now, and he's on
Okay, so-
that boulder in the background.
Oh, that guy.
That guy.
Yeah, okay.
This is just
Wide.
possibly the dead stalk in front of that species. But
It's not a
So, 'cause we
bolus.
did pass one of these earlier, and this is the other species I was referring to that has the same type of attachment as this other very common sponge, and so that's why I
Okay, so
suggested that hypothesis about the attachment type being affected maybe by the currents or the amount of
It's a
sediment
partial.
or whatever is going on in
going
this
for the
location.
base.
yeah, so
There's the base.
this actually, well, we could zoom in on the base since you're there on that coral. If you
Yeah,
can,
and
like
coming
my
up.
sponge is getting coral bombed, but it's all right. This is in the subfamily Bolosomini, but it is not in the genus Bolosoma as best we can tell. So we actually call this thing Bolosomini new genus. And we have collected specimens of it before, and it's awaiting some closer examination and eventual description. And the thing that it's kind of a weird one, but the thing that is noticeably absent on this particular species of Bolosomini is the fact it doesn't have a top osculum. It has these side things, and if he's coming around and he's looking at its face, we used to call these ET sponges because they look like two big huge eyeballs. But there's no opening on the top of the sponge like the other members of the Bolosoma genus, and so we
Be a little
really aren't
uncomfortable
sure
with that tether.
where this
Yeah.
belongs. except that it is a stalked glass sponge in the family Euplectellidae.
And its open side is facing directly into the current.
Yeah.
Okay, wide,
Thank
please,
you,
video.
Yep.
Did we get a
Full
shot of
wide.
that coral at the base, Chris?
Sure,
Oh,
there
yeah.
may have been a Plurigorgia
Around.
right at the base of this sponge. That
Yeah.
was what was doing the, what I think you said it was photo bombing.
Yeah, I think you're right, Scott. Thank you for reminding us. And Chris also mentioned that as well. I'd forgotten that guy. He's going to get in a little better position to get an image of this. And this is a
We can zoom.
different coral again. We like
Just
to
hold that and I'll
document
back
the
up.
community as we go along, trying to image different animals.
And now we can zoom some more.
I would agree with you, Scott. Do you want to repeat your identification?
Sure, I was suggesting that this is in the genus Plurigorgia. This would be another member of the octocoral family Chrysogorgiidae. We've seen many, many Chrysogorgia colonies around, so Chrysogorgia is another genus name, and genera are the level just below the family. So this is a member of the golden corals, but what's kind of interesting is that it looks white, and I think that's because the tissue is slightly thicker and reflects the light more than it does on the Chrysogorgia, which have more transparent tissues.
So wide,
And
please.
until our cruises in 2015, this was thought to be a very rare coral. Only a few colonies had been collected in the early 1900s, and I don't think they've been identified since that time. But since we've been doing all these Okeanos Explorer dives as part of the capstone expedition over the last 3 years, we've seen that these are quite common here in the Central Pacific.
Yeah, and that was the voice of Dr. Scott France from the University of Louisiana Lafayette. And last night, when we were all putting heads together to figure out a dive site on the spot, this was Scott's suggestion. So pretty darn good suggestion, Scott. Pilot, there is a fan-shaped coral in the center left coming down. I don't think we got that one yet. He's
Copy that.
keep going. He's further
are you
in the
still
back
okay
there.
with-
And I'll say that we've done
100.
pretty well with the last
Yes.
two
Bigger one.
choices.
Last would be fine.
Yeah.
Copy. 30 meters, 100 coming up.
Great.
I mean the last three
Lasers,
choices.
please.
Yeah, last three choices, right?
Right under the lasers, is that
That's it. That's the
Okay.
guy.
All right.
Actually, I'm not sure what it is from this distance.
We'll get closer.
0 meters, frame 100, speed
What
decimal 2 coming
are
up.
From a distance, it kind of looks like a Paragorgia type, a white Paragorgia. But wait till we get there.
I know. It's a strange one, Scott, at least for me. You guys might recognize it immediately, but I'm certainly not.
ROV nav bridge, we have 30
Unless
meters,
it's
bearing
totally
100,
overgrown
speed
or
decimal 2
something.
initiated.
It's off.
Lasers off,
Wow.
please.
Or Paragorgia.
Paragorgia with polyps
Good
extended
to
from
zoom.
this distance anyway.
Now, if you have a
And
minute
Scott,
for
right
CTV
you
testing.
are.
And we can have more zoom.
Yep.
So, Scott,
just
while
gonna-
he...
What you can
turn
tell
that,
is the way it sort of seems to
ground fault monitor
vibrate a
off.
little bit
Let me know if the
around the
oxygen
edges.
cleans
See,
up.
Copy.
something about the wiggle. I hope I'm not just
Well,
seeing
it's
the
going to be
video.
pretty hard to tell
But
because
You can
definitely,
just watch
I think
the numbers.
Paragorgia, you can see
Alright.
how the polyps
The
are
center window.
arising, especially from the
Alright.
tips of the branches in these little clusters. And when the polyps are retracted, those look like wads of gum, especially on the pink and the red colored ones. It looks like bubble gum. So this whole family
Starboard for tether
is
management.
This
gets
may
the
be
common name bubblegum corals, and this is a white morphology of that group. I do not know what species it would be.
Yeah, so Scott, I'm going to throw this out because I always know we have to make decisions fast. To me, this looks like a potential collection option because I don't think I've ever seen this type of Paragorgia before. Maybe you have, or Les has, or Asako maybe, but I don't know. I don't recognize it.
Is it alright to take a little more zoom?
So the one thing
Yes.
that's really distinct is the clusters where
That's
the polyps
zoom.
are arising, and I agree this doesn't look too familiar. sometimes they can fool you when they have the polyps extended versus retracted. But what I'm seeing here is these are really big polyps relative to the size of the calyx from which the polyp is extending. So I would certainly support a collection of this so we can determine if it's new or if it's just something unusual that we don't see very much.
Oops.
Yeah, Asako less 10.
I would agree. And the other,
We
I
have
think
moved about 8 meters. Do you want me to stop it maybe at 15 or something?
That's very unusual is the cluster of polyps. And then you have a bare branch area and then another cluster of polyps. And I'm just wondering whether it might not be something else other than a paragorgia, maybe an anthosalid or something. But in any case, I think the best way to know is to sample it. And so I think that getting a good sized piece of it would be worthwhile.
Let's just hold here for now.
Yeah, so pilot, we'd like probably the largest piece we could get into the box if possible.
How far do you have on the
Okay.
We have about 16 more
But
meters left.
not too large, obviously,
Yeah.
but something that'll fit.
All right.
'Cause this is quite unique in 3 years, and
So
all of the
pilot,
hurl
ship's
dives
coming
that
to
I've
stop.
been on too, the submersible dives, I've never seen
With
anything
16
like
meters
this.
to go.
Perfect.
Looks like we have a pretty good consensus.
I
Asako
believe we have all
Matsumoto from
bioboxes
Japan is
available.
also agreeing.
Is that right?
I'll just follow up on Les's comment that perhaps this is
That
an
is true.
anthofelid.
ROV
And what he's
now, Bridge,
referring
we stop
to
the move.
is the Anthophilidae
Copy, Bridge, thank you.
family, and we often see these purple corals called Victorgorgia. That's a member of that family. And both the
Gently
Paragorgiidae
out
and
on
the
the
Anthophilidae
drawer
are
because-
united in the sense that they
I'm
are
gonna
both
prep
called scleraxonians.
biobox for this.
And they have a
Okay.
skeletal
Thank
makeup that is quite
And they're
different
gonna come
from
out
the other sort of octocorals we've been seeing, the Pythagorgia
sponge
corals,
on
the
the right
bamboo
there.
corals. And because of the
be
makeup of that
effective.
skeleton, you see again the way this is kind of wiggling like it's
you want me to
light,
open all the
or
way?
it doesn't have a
Yeah,
stiff
if you
an
can.
internal skeleton. And that is exactly the case.
Yeah, I think we're all right.
Very cool. Very, very interesting animal. Sometimes we see a lot of the same things we've seen before, but right now we
You'll
are
see in
not
the zoos are
seeing
just about
something
to
that we have ever
sponge.
seen before.
I'm up against
So this is the really exciting part of
I'm gonna grab
doing
these.
this type of work here
I'm gonna
and
throw it up on
exploring
my
these
monitor.
unknown
Thank you.
seamounts.
Sorry, Chris, I'll just say as we're sitting here getting ready to collect, just behind to the upper right, maybe about 2 o'clock from the tip of this colony, you can see a very small colony, kind of looks like a straight whip. And I can't tell from here, maybe you have a better image, if that's a very young bamboo coral or if it's a little rock pin. Kind of looks like a tiny rock pin to me.
Sometimes your eyes amaze me, Scott. Yeah, you might be right on that. We're
Video, zoom out a little bit.
gonna go ahead and collect, but
Yeah, full wide is
definitely
good.
could be.
Do you want that drawer in meanwhile? Is that in your way?
No,
He's not even at an
fine.
ECC. He's not even looking on a video wall or
So,
anything. He's
Something
See
to
how
be
it
said for
goes
search
here.
image, Chris.
yeah.
I'll follow you over.
Where's my cutters? Cutters under.
By the way, I will mention that there was, I think we passed another one of these. There's 2 of these colonies together, so they were twins.
Following.
Because I think I saw both of them. The other one's a little further down slope to the right. But I wonder if we're going to see more of these as
Might
we
be a
go
tough
up
one.
too.
Gotten a little close here.
Well, that's always the uncertainty involved with exploration like this. This might suddenly become the most abundant thing
Okay,
and we don't see
video,
anything
some
else
zoom.
for the rest of the day.
Please.
Or this might be the only one
Just
we
gonna hold
see.
that.
Right. You have to sort of take advantage sometimes.
I can take that if you'd like.
Yes, thank you, co-pilot. Okay.
Tilting down.
Tilt down, yes, thank you.
I might point out that while the manipulator arm is banging around on this coral, the polyps have all contracted, and now we can see the clustered bases pretty nicely. But also note that these things have a fairly primitive nervous system, and but the signal went through the colony moderately rapidly, which is something we don't always see.
Tilting up.
Thank you.
And moving mini Zeus to the
All
box.
right, a little selective harvest there.
I can say one thing from seeing the way that it was clipped there, there's no question at all that this is a scleraxonian. So we at least have narrowed it down to one of the major groups of octocorals, and that is the group that includes the Anthophilidae, the Paragorgiidae, and some others.
Well, it's pretty exciting. So those of you watching this broadcast, by the way, you also may be looking at a brand new species to science, first time it's ever seen. We never know until we get it back into the lab and examine it. But given the number of octocoral experts that we have on the line right now and that nobody recognizes it, it's a very good chance that we're seeing something that nobody has ever seen before.
Give it a few bumps on the drawer there and see if it settles in. I don't want to abuse it too much. Oops, that's not what I meant to do. Actually, hang on a second. I can give it a little nudge.
You want me to come back out?
No, you're okay there. It's a little sticky. I think that'll suffice.
We're just getting it secured in the box.
That looks like it's in.
And we have, ta-da, it's in the box. Thank you very much, pilot.
Welcome.
Gotta be
I'm gonna follow you over to
momentarily silent here as we take a few notes.
Shoulder pull up, elbow down, wrist.
Okay, that's specimen 2, and it went.
And
again, we have all of this, the data, associated data with this thing.
Okay, finished with hydraulics.
I have a piece of that sample still on your claw.
Oh, is it?
So I'm going to throw this out while he's getting ready. Scott or Les, I don't know Paragorgia very well, or Anthophilidae. I know that Paragorgia probably have very similar sclerites to coralliids, and I know what those look like if you do a little bit of a bleach prep on them. But do Anthophilids have very distinctive sclerites compared to Paragorgids? And if I took a microscopic little piece of a branchlet there and put it in bleach, would I be able to inform you right away as to what family it belongs in?
The Anthophilids I'm familiar with in the genus Anthophila have warty spindle-like sclerites, and I would have to compare those to the Paragorgia, but I think the Paragorgia are more like spherical warty balls, aren't they? Isn't that correct?
Okay.
I'm not sure.
Paragorgia mostly has what are they called? Capstans. They're 8 radiates, 6 and 8 radiates, I think.
Finished with hydraulics.
And more similar to corallium in that respect. But yeah, I think I was just going to check on my computer, but I think that,
so Dave, you want to fly?
Guyots have these longer, larger wordy spindles, but I can send you by email a PDF, Chris, so you can have a look.
All right.
Well, thank you, Les.
Chris, you can send us the images of the stirring that you take.
Yeah, the biggest problem here that I've found, even trying to send out sponge spicules, is that even though the waters are pretty calm, when you try to zoom in on our digital microscope,
according to
very
HIPAC,
small
it should be turning more towards the
structures,
south.
very small, either spicules and sponges, or in this case it would be sclerites and corals. They're just rolling all over the place on the screen, and it's almost impossible to get a good picture. So, but I can give it a try.
I remember that when I was out there, and you have to time it with the roll.
Yeah, and I'm thinking of trying to make some improvements for the next cruise or for when the ship gets around to the Atlantic. I'm pondering this issue a bit. This is the first time I've had experience with this new microscope.
Up on top.
And so I may try to get something that might work out a little bit better and put on the ship. Okay, we are, I think, fairly close to the crest, aren't we, pilot?
I think I just came up over an edge. Maybe not the first, but definitely.
Yeah.
Popped up, then it got flat, and here's a pile of rocks.
Yeah, and looking at the HIPAC screen, it does look like we're on the top. So we are going to make a right turn, I think, once we got to the top. And I'm sure Nav is on it.
A prominent pile of rocks. It's casting a shadow. Let me just get up over the top.
I forgot that we were moving up the side to the ridge, so this will be a very interesting comparison.
Yeah. This looks like a vent of some kind, like it's a volcanic blister or something. John Smith or Jonathan Tree, I don't know if you're close by enough to the phone where you could comment while we're still here.
Yeah, go ahead.
But this could be one of these places where lava just oozed up in a kind of micro volcano here. That's what it looks like to me.
I gave my co-pilot a little tug there, so I'm going to back up. But this is an isolated blister. This feature here.
So the bridge just called up.
And we
they're
see a
doing
bunch of
heading
stuff on
change
it.
about 5 degrees. I can put in a move after they're done.
Some more of these farreid glass sponges, these upright ones that have got all this weird texturing to them. We used to call these things Skeletor before we knew what they were, after collecting some specimens years ago with the Pisces submersibles. So we've got those.
Come down a little bit here.
And those are most definitely pillow lava flows. This is how lava erupts out of the seafloor. There is a coralliate
here.
That's a precious coral. We haven't seen any fish today, so poor Bruce Mundy, we don't have any images for him, even though he can't participate today.
Okay.
We don't have any images yet of a fish for him to examine, perhaps tomorrow. There's some more stock sponges in the background, the Bolosomini. And we're doing a quick change out of pilots.
Okay, Dave, just to let you
Here,
know,
mostly just changing chairs.
the CTDs actually seem to stabilize. They have only moved from 2.37 to 2.57. That's the range I've seen, so seems to look good. If you guys want to stop doing the CTD testing.
Yep, that is what I suspected. Perfect.
Okay, and USBL also seems to be stable now.
This is sort of a beautiful corallid colony, I think, on the right side of the mound.
Let me know
if
when
it'd be
you guys
possible
are finished with the heading change, please.
Roger,
head over that way, and I
Copy,
think it's
thank you.
Dave, right? Is that right, as pilot?
Yes, we've had Dave and Dave switch spots.
Excuse me, that's
So
right.
now we have Dave as pilot and Dave as co-pilot.
Yeah, if we could get to that coral colony to the right side of the volcanic vent, it's right underneath the sponge a little bit.
I'll come down.
That fan
yes.
shaping.
Yep, I'm not sure if we have quite enough tether for that, but we could give it a try.
Yeah, it could even be the same thing we just collected. I'm not sure, but it could be. And this sponge also is something, it is attached to the substrate, and this is a really cool sponge, by the way.
Go ahead, Bridge.
We're ready for a move whenever you are.
Copy, stand by.
Okay.
So, co-pilot, they're ready for that move.
What's
are you
weird about
comfortable?
it is I think I recognize the sponge, but the
130?
sponge species
130,
usually has very, very long
30 meters.
Copy that,
grappling
coming
hook
up.
spicules on the base, and this one does not. Yet another surprise, and you see a couple of these sponges are
this
now
is
attached
nav.
to the rock.
The move is gonna be range 30 meters, bearing 130.
Let's try to come in on this.
Speed 0.2.
Okay.
That's 30 meters, bearing 130, speed decimal 2.
Good copy.
So that's a partial.
Yeah, well, it's in the genus Poliopogon. If you look at the base, it's got those grappling spicules that we've noted on other dives. And
is it alright to zoom more?
Yes.
I thought this was Poliopogon species 2 is what we call it, but these have, we used to call that species the bearded sponge because it had such a long draping of these basal spicules, but this one does not. So Poliopogon is the best I could go
30 meters, bearing
on this one.
Speed decimal 2 initiated.
Really
Thank you, Bridge.
curious now, Chris, to look back at previous dives about where these sponges are distributed. I'm noticing right here, at least
full.
relative to
Okay.
earlier on
Try
the
to
dive,
keep it
it's a little
steady,
bit
That's full.
less sediment on the rock. And you'll see as you move away from this and look to the back left, it looks like there's another Poliopogon hugging one of those upright sponges.
Yeah, and then there's another one tucked in a crevice too. So now maybe it is just a matter of larval settlement, but in this case, when this thing settled out, it was able to grab a hold of the bottom fast
Video
enough.
one should come out.
That's got a lot of fuzz there on it. And then the
Yeah, I'm
sponge
still
to the right
jerking
is
your
the
tether, but we're heading
real
there.
prize here.
The sponge.
I'm sorry, the coral to the
yes,
right.
Okay. Well, there is a sponge to the right.
I know
are
there is,
sponges
but-
and corals in every
but-
direction.
But it's not the prize,
All
I don't
right.
think. Jeez. Quit laughing at me, Nicole. Sorry. Even if I deserved it. Senior moment. That's all. I blame my age. It's out of control. Okay. I think this could be the same thing that we just collected, but we'll know in a moment. Pretty unusual sight, I have to admit.
All right, video. Let's try this. I'm way out ahead, so
Well, that's a
let's see
partial.
what we can get.
And I think it is.
I'm going to turn away for you.
And
give
there's
you
yet
some more
another
tether.
one
All right, thank
down
you.
below behind the boulder too.
Video come out. I'm going to try to land here.
Full wide.
Get a smoother shot of that.
It's going to give us a nice another look at a different colony of this coral. It's kind of nice sometimes to compare
I can't land there with that swing
a
arm
couple
out,
different colonies
so I'll just
because
get close in the water.
you have a chance to look for consistent characters. That helps with its identification from video.
Looks okay in real.
So
cam.
we had noted some rather unusual characters in the one we collected. This was Les Watling and Scott France had noted some characters, and then it'd be nice to see if the same
It's
characters
close,
appear
but
in
you're
this
still okay.
different colony.
You're good.
All right, video, come on in.
Co-pilot, we are about halfway through that move.
So is
Should
it
start filling the
this
swing
is
maybe
the same
in
thing, I believe.
2
Do you guys
minutes.
think it's the same thing? I think
That's
it
not
is.
quite full
I
zoom.
think this is the same thing, Chris. We're just looking on the other side now, different face.
All right, come in the rest of the way.
So aren't these polyps a little small for our anthothellids, Scott and Les? Because I always associate anthothelid, or I always think anthothellids have pretty big polyps.
Well, the polyps seamounts we're looking at right now look a little, well, maybe not retracted. I guess they're just a little bit limp. Looks like they're flowing toward it. But no, I did not get that impression. Certainly not on the colony we were looking at earlier.
So we're not making progress, are we? We still think it could be Paragorgia, Anthothella, Plexaurid,
We're full wide.
Octocoral, I think we can agree on. And you said Scleraxoni, I think. Is that correct, Scott?
Yeah, that would basically subsume all of what you just said. So that's, a much broader level of identification.
Okay. All right, well, great fun, something different here. And now, amazingly enough, we did not see a sea star while Chris was at lunch. So I don't know, maybe this little jinx is stopping now.
No, I think we're just, we're at a different point in the expedition, so I'm sure the stakes are just higher. We'll see, or maybe it's shifted over to you. We'll see a new sponge the next time you go off to lunch.
Speaking of sponges, we have maybe 100 in view. Are any of them of particular interest?
A lot of them are the same as we had below, Pilot, so maybe we'll...
Look in the back there. There's a huge colony in the left.
The dead one, right?
No, is it a sponge or a coral?
Well, that's a good point. I think it's
it's just out of view on the screen, and then we're coming into it.
That is a humongous dead sponge, and you are right. That is
a dead sponge.
But it's humongous. You're right.
A lot of.
Wow.
Those Chrysogorgians.
How old? We don't know how to age these sponges. I have no idea how old this was.
It's also, I apologize for distracting, because there's another very ornate Phariid. Is that what you've been calling them?
Yeah, I believe so. I think they're in.
Is that the same? See the one on the right that looks like it's got a really fancy haircut?
Yeah, I do. And we're seeing the back side of one of these things. But also there is a different Chrysogorgia going off left. And maybe some different primnoids. Okay, there's a bunch of targets, so I'm going to go ahead and leave you to take the
Which
target.
One do you want, Chris? First,
well,
Chris.
Talk to the guys on the telephone too. But
8 Chrysogorgids.
so if you swing a little bit, I mean those primnoids on the top of the pillow mound, sort of the upper left, would be something. And then if you swing further left, you see that Chrysogorgia, that very wispy one that just came into view?
Yes.
I think that's different. I don't think we've actually got that one. Okay. So those would be the targets, I think. Thank you.
Very well. Let's go check that out first.
It's hard to get at these things. It's so dense.
We need to deploy the crutch.
All right. I'm going to try to do some of this from above. Come in partial. Is this the coral that we're interested in?
Yeah, well, that's the Chrysogorgia, so that's
right.
one. And then the other one on the right is I believe that's what was the primnoid.
Okay.
So one at a time, however is easiest for you.
Okay, co-pilot, the ship has come to a stop. You still should have about maybe 5 minutes of swing.
So I
thank you, Bridge.
Throw this out there to Scott and Les if they're looking at, or I think we're slowly zooming in on this Chrysogorgid.
Try a little bit more video.
Yeah, I'm hoping Les takes the Chrysogorgia. He's better at them than I am.
Okay, I'm probably not steady enough for any more.
Sorry, I'm munching away on some food here. This looks like tricollis. Has a very wide, Chrysogorgia tricollis has an extremely wide branching pattern.
Come on out. I think
we've seen it elsewhere, also on these dives and in the Atlantic.
Okay.
Looking from
Try,
here, there's
how'd you
nothing
spell
under
the
you.
species?
I think I agree.
I put it in the event log, Chris.
Oh, very good. Thank you. Yeah, and so when
I'm going to try to get a better close-up on that.
I think that's
I have a landing point.
Yeah, that's a known species. I think the primnoid might be of more interest, actually.
All right. Well, I can maybe get them both from here.
Oh, okay. I'm anything that is great you can deliver. I'm happy to see it.
All right, let's go for the primnoid. Oh, there's. Okay. Which are we looking for here? The larger
this one,
right?
yeah. This one with all this white one with all of the fuzzy
okay
business going on.
All right, to zoom more.
Sure.
So, to our onshore coral specialists, what does this look like to you?
Yeah, I'm just waiting for the close-up here. So
I'm sorry.
It's definitely another octocoral in the family Primnoidae. So the colony that's just now off screen to the left is another one.
That is full.
Okay. And the question is whether this is the genus Narella or the genus, I think it's Calyptophora. Hang on just a second. Yeah, Paracalyptophora. And one of the things we're looking at is whether these polyps are oriented upwards or downwards. These ones are kind of standing straight out right now. And we're a little too far to see it, but earlier on, and if you continue to look at some of these close-ups of the primnoids, from a side view, you can see on the polyps, they look like rings. And so you want to try to count how many rings there are. So essentially, if you see one or two rings, it's probably going to be Calyptophora or Paracalyptophora. But if you see what appear to be 3 or more rings, probably Narella, when you combine that with the orientation of the polyps.
Okay.
So,
looks like we're...
And then you can see there's a number of sea anemones, individual sea anemones that have settled on top of this colony, and we saw that earlier as well.
Okay, and they're sea anemones because they lack the pads on the bottom? Is that right?
I'm saying they're sea anemones because the column, the body wall, doesn't appear as rough as it does in the zoanthids. The zoanthids often embed sand grains and other material, and so they appear very, very rough. And then, of course, the zoanthids are colonial, so they grow attached to one another, and you can see that these are individual polyps, so more likely to me to be an anemone, but we would have to collect.
Sounds like a closer view might help with both the coral and the anemone. Is that right?
Yeah, I think that would be warranted.
Let's see if we can
if
try
that's
that.
possible. I know
it might be possible. We'll try it. Come on out, video. And actually, we hit this with lasers yet?
Lasers coming.
Why don't we do that real quick?
Did they want a close-up of the chrysogorgia too?
I don't think so.
No,
I've seen
yeah,
that
it's it's something they've
come on
seen
in
before.
partial for lasers here. And that should do it. Come on out, and I will attempt to get closer.
So while he's maneuvering,
Scott, was that... There's a red colony down... I think it's a red colony. I think looks like Paragorgia on the lower side. Is that something you're interested in as well?
Yeah, on the lower right there, that kind of looks like what we were saying was a coralliid.
Oh, okay.
Earlier,
so we should definitely go take a look at that also. Okay.
Sirius is looking southeast, and it's pretty clear in the Sirius view that all of the growth is on the southwest, the upcurrent side of this feature, and there's nothing on the downcurrent side to speak of.
Is any of the surveys, watch leads, listening to you?
They're probably not listening to me, and I
Alright, video.
can repeat that on it.
All right. Should I just come all the way in?
Yes.
Okay.
Okay, Scott, we have a max zoom coming right your way.
Let's see if there's part of it that might have a darker background.
Yeah, so you can see how rough I'm talking now about the smaller polyps.
That's better.
And they look rough on the outside of the column, the body wall, connecting to the main branch. And hang on a second. And so yeah, let's see, I'm trying to find the right ones. And yeah, you can kind of see what look like rings, and they're not really rings. It's that the polyp has sclerates that are made of scales, almost like a suit of armor that knights would wear. Mm-hmm. So the ring is actually the flange of that scale. And in a Narella, there would be 3 or 4 of those scales, counting from where it attaches to the branch, going to where you see the tentacles sticking out. Whereas in Calyptophora and Paracalyptophora, there would only be 2. And then it gets complicated because then there's also some opercular scales right around the tentacles. These, it looks like there are more than 2, so I would say that this is Narella. And then you can also see where the tentacles come out. They're kind of nodding down a little bit. And I think I am still going to stick with sea anemone as a guess for what these other polyps are that have settled on top of the coral.
Yeah, they look almost kind of actinoscyphid-like, I think. So are you guys good, Scott?
Absolutely. We can move on.
Very good. So let's take, Pilot, let's take a look at that. There's a pink colony in that valley to the right, which is fairly large and kind of branched out.
I think I see it.
Yeah, that's it.
Nips. That one?
Yes, sir.
There's no way I can set down here.
You know, there's another one on the left side if it's easier. On the other side of this rock, I think there's a colony just like that. I don't know if it's any easier to reach. It's a little farther down, I think. Yeah, do you see it?
No.
Oh, okay. So there's a sponge, or there's a sort of a pink, big tree-like thing that's right next to one of the bulb sponges, right in the center to the right.
Oh, okay.
Yeah, and I think that's the same kind of coral. It looks like there's a clearer
that is a lot easier
platform to reach. Yes, it does appear to be a bit more accessible. And it's bigger, so there's more stuff on it.
Might be a new bamboo coral also on the far upper left after you finish with this one.
Oh, wow, okay.
Come on in. Okay, now we're settled in.
Now, if I'm not mistaken, this is a precious coral.
As much as you like.
Is that hexacorallium, Scott?
It's not quite full.
Chris was suggesting hemicorallium earlier.
Hemicorallium, my apologies.
You are correct that it is one of the precious corals. Wait for the zoom and the focus. Yep, I think that is fair. So it's certainly another octocoral. It's in the family Coralliidae, which are the precious corals. And I personally can't say for certain if this is corallium or hemicorallium, but I've been hearing hemicorallium an awful lot, so I think that's a reasonable guess.
Okay, that sounds great.
The tentacles are somewhat withdrawn, or at least the tentacles are kind of curled up. The polyp hasn't withdrawn into the seamounts.
Okay.
Which is the name for the tissue that covers the main branches.
Interesting. So, okay, we can move on. So, Scott, tell me where this bamboo coral was, the one you said was to the left?
Sure. Yeah, it's going to be left a little bit further forward, the direction you were going. And very
you mean
wide
these?
Oh, wow.
Yeah,
that
okay.
is big.
Ah, how did I miss those?
Yeah, it's pretty big.
Struth.
Hard to miss.
Yeah, so I'd say the biggest one, or whichever one's easiest, is probably...
Nav, let's go.
Whichever is at your pleasure.
Are these
Yeah,
two the
it's
same?
that one on the left. It's different from
Let's
the
go
one on
20
the right.
meters
Yeah, they are
at
different.
0,
Actually, yeah,
0.
the yellowish one on the left, I think, is the
Actually,
one we want to
belay
look at.
that 0. Let's see here.
Because I think the one on the right is the. I'm sorry. Yeah, you just said that. Yeah.
20 meters at 115, please.
Bridge, this is Nav.
Coral looks nervous.
Go ahead now.
Let's get a move in. That's
Alright.
gonna be 20 meters, bearing 115, speed 0.2.
20 meters, bearing 115, speed decimal.
So that's a partial.
Huh. Well, it certainly looks the skeleton looks like bamboo coral, Scott. But
Polyps.
why are the polyps all withdrawn?
Yep. The polyps are all withdrawn. Something has
ROV now bridge move
disturbed
of
this.
20 meters, bearing
And
115,
they
speed
are
decimal 2
somewhat
initiated.
mounded, and you can see that
Oh, there's one section with the polyps
branches
out.
are arising
The
from
crinoid.
between the
Yes,
dark
right.
stripes there. Those stripes are nodes made of basically a protein,
so there's some missing
and between
tissue
that we refer to it as
below
the internode. So this is an internodal brancher, and the shape of the polyp suggests that this is probably in the D2 clade and perhaps in the genus
theory out here.
Ecnomyces, which is a genus that Les Watling and I described several years ago in the Atlantic. So I'm not sure. Oh, there we see one branch in the center where the crinoid is. The polyps are actually extended on that branch. I'm not sure why they have retracted on all the other branches.
Yeah, that's
Chris,
really
can
weird.
I throw out some
Colony,
crazy
pretty old,
speculation?
and I'm basing that on the diameter of those lower branches. Go ahead, Chris.
Go ahead, pilot, please.
There is no tissue at the base of that branch that has the polyps extended. I wonder if it therefore lacks whatever form of communication it has with the other branches.
That's interesting. So, Scott, the pilot suggests an interesting notion that...
Just heard what the pilot said.
Oh, you did? Oh, so...
It's a fantastic observation, pilot, so thank you. So Les before was saying that they have a primitive nervous system, and actually it may not be so primitive now. There's a lot of studies going on the genetics of these cnidarians. But at any rate, the polyps are able to communicate with each other, and if there is no tissue on the base of that one branch, then it's not receiving the communication from the other polyps, and so it would have no reason to retract, which suggests that whatever the disturbance was affecting some other part of the colony. So, pilot, fantastic observation, very nicely done.
My hats off to...
I'd like to add to that. It looks like there's a tissue break on this branch to where the polyps are still extended. You scan down slightly, it looks like the tissue's missing from part of that branch. And right there. And so that would break off the nerve connection. All these cnidarians have, the nerves are in what's called a nerve net. And there isn't like a central nervous system or whatever, but there are nerves that extend all the way down the branches and then up to the polyps. And a number of years ago, when Scott and I were working in the Atlantic, we did a really neat experiment where we reach out with have the pilot reach out with the manipulator arm and touch the side of the colony, and you could watch the wave of conduction of the nerve impulse go down the branch until it met a junction with another branch. Then it would go up that branch. And so it would take a little while, but sometimes 15 or 20 seconds for that to happen. But here it's clear that the tissue connection is broken. And so this is probably why the polyps on this branch are still open where they're all closed on the other branches.
Wow. Are you guys happy with the image so far? Do you need to? What other aspects would you like to look at?
This is good for us, Chris.
Okay.
This looks to us like D2 clade, possibly likely ecnomyces.
Okay, co-pilot,
and I'm
the ship
basing
should
that
be coming to a
mainly on
stop
the sort
soon.
of volcano shape, that low mountain shape of the retracted polyps. So it's kind of helpful that they were retracted.
Yes, definitely a...
I wonder also if it's possible to get a can we get any closer on those polyps that are open so we can see what the sclerite arrangement is like? They're kind of small, so it might not be possible.
Yeah, let's see what we can do.
Yeah, come on out.
They're about to come out, and then they're going to zoom in.
Wow, nice one. I came in at the right time, didn't I?
Well, we had a very interesting observation too. That
ROV nav bridge move complete.
That's Dave up there, isn't it?
Thank you.
Yeah.
Bridge.
That Dave made. That there's only one branch that's actually got the polyps all
Other side,
open.
maybe?
The rest of the colony is all closed. The tissue is absent from the bottom of the branch.
Every tail
and
well,
so
that's
Dave
gonna be tough.
opines that it might be that they are not communicating with the rest of the colony. And Les and Scott agreed emphatically and sort of explained that whole process. And so we're now trying to look at the polyps and see what type of animal it is.
Is that full?
That's full.
Okay.
It's also interesting to note.
Oh, yeah, that's great. I think that an actomyces relationship is pretty strong. You see there are no big needles between the tentacles, and the rods that you can see in the body wall of the polyp are all at an angle, and that's pretty typical of actomyces.
Oh, great.
And so when we're done here...
Thanks for that shot. That's fantastic.
Okay. So we have a request from Asako, when we're done with this, to look at the precious coral colony on the right.
Okay.
So when we're done with video, we can pull out and take a look.
I also noticed there's a holothorian right behind.
There is?
Yep, right at the
What's that?
middle top of this coral.
What made you go, ooh?
The rocks behind it.
Can we look through this? Or
I can try to.
Oh, okay.
Where is it?
It's right there.
Ah, okay.
Well,
sneaking through.
Yeah, let's take a look at the precious coral, and then we can sneak around and see what else. Let's see if maybe that sea cucumber is not too busy. We can ask it some questions.
So that sea cucumber probably goes by the name of Hansen to its friends. But
interestingly enough, that's the name of the the surname of the person it's named after. So.
Okay, come on in. We actually have several corals in between us, but I think we can get it from here.
That's a partial.
Oh, there's a cute little sea anemone down there too, I think.
All right, let's come in tighter on this right side.
Yes.
So there's there's really only one hemicorallium, or a member of that genus in the Coralliidae that I can identify so far. Beyond hemicorallium. Hemicorallium's pretty easy to differentiate from the other two genera that have recently been sort of accepted, the corallium and the pluricorallium, and I think that's pretty well accepted right now. But most of the time all we can do is just hemicorallium and all these things. I think there's a hemicorallium lauense, a hemicorallium near lauense, hemicorallium I think regal, I guess. Abyssal is the one that is distinctive enough where you can actually do something with it.
Nope, that's max.
I think we're good when you're done video. Thank you.
Yep, video's good.
You're good. All right.
And we're full wide.
Okay, well, let's go jump over and take a look at that sea cucumber, and then maybe if we
Last
could
move I made
spin
was
back around and
115
look at
just
some of
to
those
come
big
to
sponges
you.
that we passed up.
To get this very flattering view of you at work.
Oh, thanks for that.
And I defer to Nav
So
to the
is that him, or
greater
is that a different
picture
one down
of
there to the-
where away.
That's it.
Yeah,
Right
that's the
there,
one. That's the purple
waypoint
one
2
on
is
the left
selected,
there.
so the bearing from the ROV is
I'm
155.
not absolutely sure he goes by
155.
Hanson.
Okay, you
We
can
will-
go ahead and call in a
You
30-meter
should.
155
We
move.
should probably be thinking about waypoint 3 at this point.
Yeah, that's what I meant, sorry.
Okay, great.
Oh, well, there's so
I'm just
many
going to look
stops
down
by.
here. So this is where you've been. You worked your way
Yes.